All right. So today we are going to continue on our theme of population density in this in our unit, and we'll focus on population ecology, which is the study of a population's vital statistics, how that population interacts with the environment, and then those interactions within and between the various populations in that environment can influence the survival and reproduction of those individual members within that population. So ecologists then go into the field to test their theories about those species interactions and population dynamics, and also to monitor the health of any threatened or endangered populations. So in today's lesson, we'll study and look at the ways in which scientists measure those population changes by examining the dispersion patterns and by determining the size and density of the population. What we'll learn today is terminology related to population dynamics. We'll learn to describe the characteristics of a given population, such as its growth, density, fecundity, mortality distribution, and minimum viable size. We'll take a look at how population density is calculated, and also how to estimate total population size, both directly and indirectly. What are the characteristics of a population? Well, we discussed what a population is even in our last lesson. A population is defined as a group of individuals that are of the same species. They occupy that same area, they interbreed. And every population has got some specific demographics, or in other words, characteristics. And those are things like the size, the density, the distribution, and the number of individuals of various age groups. And population density is very important because scientists must measure or estimate this population size in order to calculate population density, and then to be able to make predictions about the future of that population. In order to calculate that, we need a few factors. We need to know the population size, which is the number of individuals that can potentially or actually contribute to the gene pool. Excuse me. Population density is represented by D. Excuse me. I'm sorry, I'm just going to mute for a minute. Guys, just bear with me for a moment while my cough calms down. I'm sorry, I apologize. I'm back. I think I'm done, hopefully. So, right, population density given by the symbol D is the size of the population within a specified area or space. And you calculate it by dividing the number or the total number of individuals of that species divided by the area that is occupied by that population. So the total number of individuals in the population divided by the area occupied by that population. Let's go through an example. And I apologize that this equation earlier on, I noticed had reverted into something kind of hard to see, but I think I tried to fix it. I hope everybody understands that population density is a total number of individuals n divided by area occupied by the population, or S. So as an example, the population of deer living on a 300-hectare natural reserve is called, is 225. So 225 deer living in an area of 300 hectares. How do we calculate population density? You just divide that population divided by the area that that population is occupying, and you get about 0.75 deer per hectare. And there's two different types of population density. One is a crude density, and the other one is the ecological density. The crude density measures the number of those individuals in the same species within the total area. And the difference is that in ecological density, you are measuring the number of individuals within the area that is actually used by the population. So there's a distinction there between crude and ecological density. So in that example that we just talked about up here with the deer, there were 225 deer living on 300 hectares of land, and that would make the crude density 0.75 deer per hectare. However, if we maybe take a closer look and we see that these deer are actually only living on the southern 100 hectares or so of that preserve, and maybe never even go into the other 200 hectares that are further in the north. So in this case, the area that is actually being used by the population is only 100 hectares. It's not the full 300 hectares. So the ecological density would then be 225 deer living on 100 acres. So the ecological density would be 225 deer divided by 100 hectares, or 2.25 deer per hectare. So there's a distinction there. And then different species that are occupying the same area typically do compete for food and space. Just a second, Ahmed, I see you as joining twice. What's going on?
Yeah, I'm just switching devices because this one ran out of charge.
All right.
Thank you.
That's fine, no problem. I'm just gonna continue then. So you have different species that would be occupying the same area and competing for the food, competing for space and all the other resources. And these interactions between these species will generally influence the population density and also the dispersion of that population throughout that habitat. And populations of organisms will typically follow 3 different patterns of distribution, of how they are distributed or dispersed within that area. And those are clumped distribution, a uniform distribution, or a random distribution. And here we're going to define what do we mean by these, but also this figure just represents, just pictorially shows, and so we can have a better understanding of what these terms mean. In clumped distribution, which is the most common form of population dispersion, you have species that are able to be responsive to the conditions and resources which are often patchy within the habitat. So they might clump together in certain areas within that habitat. For example, you might have schools of fish clumping together in certain areas, maybe closer to the coral reef or closer to an area where there might be a habitat for them within, maybe within some sea kelp or some within a reef. The second type would be a uniform type of dispersion, such as these penguins that are just looking like they're uniformly distributed out here on this area. And here organisms are very evenly spaced out, and very evenly spaced out, much more so than you would expect just by random chance. And this generally happens when there is competition for resources or space. And also the animals are very territorial. So it happens when there's territorial behavior in the animals, there's competition for resources or space, so they will kind of spread themselves out enough in this uniform type of dispersion pattern. The third type is random dispersion. And this isn't very typical. It really happens when there's conditions that are almost uniform and resources available, and the population neither seems to attract nor avoid one another. And this is typically a pattern you might see within a deciduous forest, for example. The trees are spaced out, not exactly uniformly here, but kind of in a random pattern. So how do we then calculate, actually calculate population size? There's a number of different methods used to determine the size of individual populations. And those are the direct method, indirect method, or to estimate the size. In direct method, you go and count all the individuals in the population by hand. And this method is quite time consuming, if you can imagine, especially if it's a large area with a large population. It's also impractical or sometimes impossible if the number of individuals are so high or if the area is so large. The second method is indirect, and scientists use this method of indirect observation to get an estimate on the numbers in a population. For example, they may use the number of fecal droppings or the number of tracks left behind by animals, or nests or burrows that they found to estimate how many individuals are in that population. And then there is other methods that can also be used to estimate the size of a population. For example, if you have an organism such as a tree, which is immobile, trees don't uproot themselves and move around. Once they're planted there, they're quite fixed. if you have this type of a situation, that they're immobile and they are fixed in their position, scientists can estimate their population using a quadrat sampling technique. And quadrats are these sampling areas of the same size and shape. For example, a rectangle or a square, sometimes even hexagon type quadrats are applied. And for example, you might have a quadrat here of 0.5 meter by 0.5 meter. And within that, you might have squares that are 10 centimeter by 10 centimeter. So you can use this as a way to help estimate the population. So the number of individuals is counted within some of the quadrats that are being sampled, and then an average of individuals per quadrat is calculated. And then this number gets multiplied by the number of quadrats that are within the total area that is being estimated based on the number of individuals. So this is another method to use a quadrant. Another method is a mark and recapture method. And this is useful in the opposite situation where the organism, the individuals in that population, are very mobile. And for example, in bird populations. And in this technique, you sample a number of the organisms, some of the individuals within the population, and you mark them in some way. For example, you can see this bird has got a tag put around one of its claws, and they get tagged, and then they get released again and allowed to mix within the rest of the population. And after a defined period, a set time, then in the field you go and you recapture another sampling of that population, and you see the proportion of those marked individuals to unmarked individuals, and you use that to estimate the size of the population. And in order for this method to actually be somewhat accurate, some assumptions have to be made about this population. And these are that the chance of being caught is equal among the individuals. And it is not, for example, during a period where females might be in their burrows or anything like that. So you have to make sure that, in fact, all conditions are such that all members of the population are equally represented and are at equal chances of getting caught. You have to make sure that there has been no rapid change in the overall population, either due to some disease or death or immigration or emigration. Of course, unless you have already established the baseline number of this population, and now there has been some event as an immigration or some disease or death, and now you're trying to follow that change. But otherwise, when you're establishing your initial population size and want to take a good look at this, you want to make sure that there's been none of these events influencing the population. And you also have to allow for enough time before you have the recapture, so that those animals have a chance to adequately disperse and mix in with the rest of the population. And also you have to make sure that the markings are in such a way that they're not lost during the duration of your study period. For example, if you have an endangered bird population that is being monitored, and scientists go and mark 30 birds and release them back into the wild. And 6 months later, so 6 months, this is your specified time, researchers go and recapture 50. 50 birds. And they find that 20 of these 50 birds have their tags on them. So in order to estimate what is the total bird population, you can use this type of a relationship, this type of equation, where M is the total number of marked organism, N is the total population. And so the ratio of total marked organisms to the total population would be equal to the number of marked organisms that are recaptured, or small m, over the size of the second sample, or the recapture, small f. So you would, and this makes sense that this ratio should exist. So you can use this relationship to calculate the total population, or N. And so N would be equal to uppercase M times lowercase n divided by lowercase m. So the total population would be the total number of marked organisms multiplied by the size of the second sample that was recaptured, divided by the number of marked organisms that were recaptured. So in other words, 30 birds were released, were tagged and released, times 50, this is the recapture size. And then of that recaptured size, 20 had the tag. And if you do the calculation, this will suggest that the total population size is about 75 birds. And there's some homework questions. So this population dynamics that we've been talking about, it studies both short and long-term changes in size and the age structure of the population. And it helps to identify both biological and environmental processes that have influenced those changes, both in size and age structure, and studies in what way populations are affected by birth rates, death rates, and by immigration and emigration. In measuring population changes, demographic variables that determine increases and decreases in population size include things like the number of births, deaths, immigration, emigration, and also the type of species and its fecundity. So by births, or natality, this is the production of new individuals via birth or hatching or germination or cloning. If you're looking at fish or birds, it's how many hatch. If you're looking at seeds or plants, how many germinate. If you're looking at maybe bacteria that you're cloning, and it's the number of clones. And by deaths or mortality, this is the lifespan and the percentage survival at different life stages. And by immigration, we mean introduction into a population, either by flying in or swimming in or walking in or floating or hitchhiking on other animals. Or there could be emigration, which is the dispersal or movement out of a population. And also depends on fecundity of that type of species, or its potential to reproduce offspring in one lifetime. For example, a rabbit has a much higher fecundity than a human. In their lifetime, rabbit produce far more offspring than human. And then there are three general patterns for survivorship in species, shown by this survivorship curve here. The first type is represented here by the red line. Type 2 is the blue, and type 3 is the green over here. And in type 1, what you have is often a population with very low mortality, long life expectancy, and typically very slow to reach maturity, and they often produce a small number of offsprings. For example, humans or elephants are good representatives of type 1. So as you can see, on the y-axis is the number of survivors in a log scale. And in the x-axis, we have the age in relative units. So as you can see, the population, in this population, the individual, the young are slow to reach maturity. It takes a long time to reach maturity, and have very long life expectancy. As you can see, the age goes on for a while. Most of the population is in this area up here. And then very low mortality. So not a whole lot of the population is in this area. As the age goes up, there's less and less of that population in that area. In type 2, and this is very representative of birds, you have uniform risk of mortality throughout the life. So from the time you have an egg and it hatches to all the way to, it's very uniform. And in type 3, you have very high mortality rates when they are young. Typically there's large number of offspring, and this is very typical of plants. Lots of seeds are made, for example, and a large number of offspring, those being the seeds or the acorn, for example, that may fall from the oak tree. And but on average, there's very low life expectancy, and another good example of these are also like plants like dandelions, or really any kind of a plant, but also in the animal population, a sea turtle. They produce lots of offspring. They have very high mortality rates. The young turtle, when they first hatch, before they go back into the water, quite a number of them die off very quickly. And then a few that live on will live on to an old age. So the other important topic of relevance when we're talking about population dynamics is the loss of species and biodiversity. And many scientists think that currently we're living in another mass extinction that is similar to the time of the mass extinction that took place at the time of the dinosaurs. And they worry that by the year 2100, that 25% or maybe even more of all plants and animals that were on Earth back in 1900 will have become extinct by this age. And if mass extinction is underway, this is taking place as a result of human actions. And the term biodiversity then means the number and variety of the species that are that exist on Earth. The number of known species is actually nowhere close to the actual number. And there's many parts of our planet that are still undiscovered, and scientists know that there are many species of plants and animals in these locations, in these undiscovered areas, that have not been identified. And there's estimates of the total number of species on the planet range anywhere, have been estimated to range anywhere between 10 million to 100 million total number of species. And it's human population growth that is causing this dramatic and rapid change to the environment. And some of these changes are happening so quickly that plants and animals don't have enough time to adapt. And as a result of their environment being affected, their habitat being taken away, they become extinct. There's many reasons for extinction. Main ones are habitat destructions, of course. There's also unregulated hunting and poaching, and also the introduction of foreign species into the ecosystem that come in as invaders and take over. So by sustaining biodiversity, it helps to preserve the ecosystem. It's important to maintain a healthy ecosystem because that's a good way to ensure a healthy biosphere with a balance in the flow and cycling of nutrients. And in such a balanced ecosystem, each species has its own role to play and makes its own contributions. There are species that depend on each other for their survival, and some species play such an important role, such a key role, that they are called keystone species. And an example of a keystone species would be the sea otter. And the sea otter is the story of the sea otter is that the sea otter lives on the Pacific coast of North America, and they feed on shellfish and sea urchins that live among the coastal kelp beds. And back in the 1800s, sea otters were trapped because of their fur until it was thought that they were extinct. And once the sea otters were removed from the ecosystem, from overhunting to the point of extinction, this really led to a very negative and detrimental sequence of events. With the removal of the sea otter, which was essentially the sole predator for the sea urchin population exploded exponentially. And since sea urchins feed on kelp, the kelp beds declined and were decimated, which in turn was very bad for all the other aquatic organisms that live among the kelp. Now, since then, the sea otter has been reintroduced into the area, and as their population has come back, the seabeds have also returned and balance has been restored to that ecosystem. So that is a story of the sea otter as a keystone species and how they play such an important role within that ecosystem. And Canada has a Species at Risk Act, which is a very small step towards protecting Canadian wildlife. And currently it only applies to federal lands such as national parks. And what this means is that most public or crown lands in Canada that are under provincial jurisdiction, and only a tiny fraction of Canada is under that direct protection of the Species at Risk Act legislation. And this legislation does contain some special provisions that can protect areas that are normally under provincial jurisdictions, but these apply only under some very special circumstances. And this protects species that are designated as at risk. That's what this is. But there are currently 487 species or so on the species at risk list. But currently SARA or SARA only protects 345 of them because the Act excludes species if protecting them may cause economic or social impacts. And these types of exclusions actually go to weaken the effectiveness of this act. And because it is usually economic considerations that have put these species in harm's way in the first place. So yeah, a lot of work still needs to be done in this area of preserving species that are at risk here in Canada. And that brings us to the end of our lesson. As promised, it's a very short lesson today because I wanted to make sure we have enough time to hear from our presenters. Okay, so today, who do we have? Okay, I'm just going to take a quick attendance. And I see Ahmed's here. Mana's here. Natalie. Here. And Patrick is here. Yes. Okay, perfect. So we have already heard, excuse me, from Ahmed, Natalie, and Patrick last class. Mana, do you, I'm going to turn on my camera. I'm going to, where am I? I'm going to stop sharing. Hi. So Mana, if you're ready, you go ahead and turn on your camera and share your screen with us, and we can hear your presentation. Are you ready to present today, Mana? Mana? Okay, so okay, I guess Mana is having some problems. All right, so we can conclude our lesson here today. I hope we have enough time to hear your presentation next class. And I hope that everybody else who still hasn't presented yet will be also able to present. All right, guys, we are getting very close to the end of our course. Let me just remind you guys of where we are. Okay. All right, I'm just going to share a screen with you again. Okay, so we have yeah, we are at the end of our unit or the last unit, unit 5, population dynamics. So today we have finished lessons 17 and 18 up until this point. So our unit 5 assignment, which I will post tonight, is due Sunday, December the 4th. Again, each one of these assignments is worth 10% of your mark. Please don't leave 10% of your mark on the table. Please hand in your assignments, every single one of them. Claim as many marks as you can. Answer all of the questions. Please don't leave anything blank. If you leave something blank, I have no way of giving you any marks for it. If you at least make an attempt at answering it, perhaps you might receive partial marks. So yeah, I just wanted to make sure everybody knows, these dates are not correct. No, they are, sorry. Yeah, so yeah, November 28th and 30th, we've got 2 more lessons to go. And then I will hold a tutorial session for you guys on the Monday, just before our exam, and our exam will be held in person at the academy, just like the midterm. And I think I'm okay to make it this time. I hope I don't get sick again. And I will be there also to help you guys if you have any questions. On Wednesday, December 7th is our exam. 2 more lessons left. We're getting very close to the end of our course. Please submit any unfinished assignments or coursework, whatever you have, please submit them. I can't emphasize that enough. And thank you for all of you. Most of you have submitted everything, and I do appreciate that. Good for you. And all right, in that case, I'll see you guys next Monday then. Take care.
Thank you. Bye.
Okay, have a good night.
Bye, Miss. Have a good night.
Have a good night, Miss.